Hi, my name is Julia Dugan, and my project is studying the effects of interferon alpha-2 seeds enclosed encoded nanoparticles on HIV prevention. So just a little background, interferons are small cell signaling protein molecules that are released from tissue cells when infected with a virus. Now, some common issues with interferon include its short half-life and its inability to stay in the bloodstream. Now, this brings me along to my approach of nanoparticles. Now, nanoparticles would be ideal because it can be stored as a dried powder for a long amount of time, and with this new drug approach, the interferon will be able to effectively aid in antiviral activity, as well as increase the amount of time it's in the interferon is in the bloodstream. So essentially, the goal of this project is to optimize this novel procedure. Now, so moving on to the methodology, first I prepared 3 samples of light nanoparticles, interferon enclosed nanoparticles, and interferon-coated nanoparticles. So first I hydrolyzed TMOS, which is also known as tetramethyl orthosilicate, and upon hydrolyzing the TMOS, I then prepared 2 samples containing chitosan, PEG 400, and 50 millimolar phosphate buffers. In these 2 samples, one sample I added the hydrolyzed TMOS to and allowed to sit for 15 minutes to hydrolyze, to polymerize. The second sample, I then added a sample of 50 micrograms per microliters of interferon into the sample, then added the additional TMOS and allowed to sit for 15 minutes. Upon the 2 samples polymerizing into a gel, I then placed both samples into a lyophilizer, as shown here, where I then placed the resulting gel into a lyophilizer known as a viscous lyophilizer for 72 hours in order to dry from a gel state to a powdered state. Upon drying, the powder was then ground up, and the blank nanoparticles were placed at room temperature for storage, and the interferon enclosed nanoparticles were stored at -80 degrees Celsius to prevent degradation of the protein. Now, the third sample that I prepared was interferon-coated nanoparticles. So this sample was actually prepared on ice, again, to prevent the degradation of the protein. And first I took a sample of the previous blank nanoparticles created. I washed it in PS solution, and I set it to incubate with 50 micrograms per milliliters of interferon for 1 hour at 4 degrees Celsius. The final sample and the supernatant were then stored at -80 degrees Celsius. Now, in order to test that the interferon coated the surface of the nanoparticles, first I placed 2 samples through an FPLC system, which is known as a fast performance liquid chromatography system, for 1 hour. The first sample that I placed into the column was 0.2 milligrams per milliliters of pure interferon, and on the second column I placed a sample of the supernatant from the interferon coated nanoparticles supernatant. Bringing your attention down here is showing the results of the samples. So as you can see, at 30 minutes, there's an absence of a peak for the interferon nanoparticle supernatant. This shows that there's actually nanoparticles present in the columns, which means that the interferon has effectively coated the nanoparticles. This means that I was successful in coating the actual surface of the nanoparticles with interferon. Upon establishing that, the sample was then again placed in -80 degrees Celsius for storage. Moving on to the second phase of my project, which was establishing a concentration gradient. I used various concentrations of the nanoparticles because the nanoparticles combined with the interferon, I did not know how it would affect the HeLa cells that I plan to use. And the reason I plan to use HeLa cells was because they have been previously used in different studies for different viruses, such as the polio virus, as well as the vaccine. Also, so upon establishing a concentration gradient, as shown here, between the concentration of nanoparticles as well as the concentration of interferon based on each sample, I then prepared samples. I prepared the samples into three stock solutions, three concentrations of stock solution into three categories. I then, okay, sorry. So I had four different samples. I had a control sample, which did not get any nanoparticles. I had a blank sample, which just got administered the blank nanoparticles. A sample containing the interferon enclosed nanoparticle, and a sample containing the interferon coated nanoparticle. The laws were, so what I did was I exposed the HeLa cells, I exposed the HeLa cells to the interferon as a pre-treatment for 6 hours. Upon the 6th hour, I then replaced the sample. I replaced the media with fresh media and administered the genetically engineered HIV sample, and that was allowed to sit for 72 hours. After the 3rd day, the samples were then photographed under a UV lamp. Now, what's unique about this HIV sample is that it contained a GFP protein, which allowed observations to be done under the UV lamp, showing contamination. As for the results show that the interferon had no effect on the HIV samples. Pre-treatment, So this shows that there may have been some inconsistencies. First of all, there may have been problems with the surface receptor of the HeLa cells. Maybe it did not recognize the interferon cell receptor. Also, what I observed was that in the higher concentration sample, I noticed that most of these cells were very weak but not dead, and there were also signs of contamination. All of these factors may have contributed into why the interferon did not work, and I would like to further study an alternative of how to make these nanoparticles work on these cells. Another approach I would also like to use is study the topical use of interferon on other viruses such as herpes simplex.